Just 2 words. I mean, obviously, Patrick gave a nice introduction of our today's speaker last week because he gave the Rothschild lecture last week, and so I don't have to add too much. I can just add a couple of personal words. Well, we're just talking a little bit about the past, but in the early 90s, I took a course with Chris Klaassen, and then I got to know about Peter, and then in the mid 90s I visited Berkeley as a prospective PhD student, and you very kindly showed me around at that time. I decided to move to Chicago. Sorry, I didn't. But nevertheless, Berkeley was very attractive. And since then, obviously, well, it's very pleased to see that Peter has moved into the field of networks as well, and along from semi-parametrics a long way maybe, but clearly a very productive statistical scientist and very innovative. And today working on something that is called network modeling of topological domains using IC data. So very pleased to
thank you very much. Well, Carrie Priebe will tell you that actually semi-parametric models may in fact be coming into network modeling as well. In any case, yeah, well, I committed to this lecture.
It's only number 3.
Yeah, I know. And I relied on my collaborators, and they delivered. But it's an applied lecture, and I'm not really an applied person. I just tag along. So this is going to be a short lecture, but I think it may be fun anyway. So this is really work done primarily by Rachel Wang, who was a PhD student at Berkeley and is now a Stein Fellow at Stanford, and then going back to Australia. And Purnamita Sarkar, who was a postdoc with me, who is now a faculty member at Texas at Austin. And Anshul Kundaye, the last, is, it's usually in biology you have the people who read the work, and then at the end, the formal sponsors. And Anshul plays somewhat that role too. He actually was a postdoc on the ENCODE project, but he's now an assistant professor of genetics at Stanford, and it's his data that we're working with him, but he's very actively involved in the modeling as well. So let me start. So here's the outline. I'll first of all, talking about biological background and motivation, and it's a sort of subtle kind of data, so complicated in some ways. So I'll spend some time on that. Then, and then the model, which is it's a network model, all right, but it's more complicated than just a network model. And the results, which are promising so far, but as far as I'm concerned, they're not conclusive yet. One of the problems, as you'll see, with this investigation is that it's a very hot area. So the papers are 2015, 2014, and there are people working on this all the time. So in particular, that's an excuse for not having any theory. Okay. So, to start with, you all know about the genome. And the genome is 2 meters long. These molecules. And I forget always how many we have. I think it's been revised, but what is it, 36, 38 chromosomes? I don't remember. But each chromosome is a molecule. But this is all packed. I mean, copies of these things are packed in each of our cells, and our cells are little boxes which are a few microns wide. So you have to think, how the hell does this stuff get in there? And it's tightly packaged, but it's also accessible to the transcriptional machinery. The transcriptional machinery is really, we talk about the genes, but somebody has to tell the genes what to do and when to do it. And so there's all sorts of various things in between the genes which do that, which have that function. And what this implies, by the way, is of course that most of the chromosome is actually not accessible in any given cell, right? Most of it is still tightly wound around its spools. But there is a structure which is now being investigated by some very, very clever experiments. So here is the structure. The DNA are the molecules that we know about. They wrap around things called histones, which are these things, right? These spools, and these are proteins, and then they're form a string, and then you go around a loop, and then you continue. And the central part is each of the histone block, if you want, is called the nucleosome. It's not too important. Now, in order for things to work, right? I mean, they have to be accessible, and so the chromosomes really look like that, rather than these nice strings that we've all seen in our biology classes. They're little balls, and they are packed into the nucleus. You can, I think, quite clearly see actually why crossover occurs. It's a wonder that more doesn't occur, given these structures. But I think there is a process by which they straighten out when pairing off, of course. And you have, and again, these are governed by these histones. You have megabase scale, so you have this folding and looping on a kilobase, 1,000 bases, or million base scale. And this folding and looping is within these things called topologically associated domains, which will be defined on the next slide. And then besides that, there is this million megabase scale actually active and inactive parts of the chromosome in every cell. And the chromosomes have discrete territories. Okay, so what are TADs and why are they interesting? Well, they're contiguous regions of enriched contact and interactions separated by actually boundaries that you'll see. So it's essentially, you can think of a TAD as a region, as an area where things go on, right? There are genes, there are gene regulators of different kinds, and then they're separated by things which you'll see in a second, which are called, that's on the next slide. Okay, so they have key roles in the regulation of gene expression, and the loops you see are actually permit parts of genes and their regulatory elements, like promoters and enhancers, to be close to each other. I mean, that's one of the interesting things when people first looked at the genome. They said, well, there's proximal elements and distal elements. Well, it's a wonder that any distal element does anything, because it would have to go a long distance. That protein would have to go a long distance and to get to its destination. But in fact, they're not far from each other, not in 3 dimensions. And that's achieved by this looping. So the packaging has an important role. Okay, and the evidence that these are separate units, they've been identified to some extent, is that the gene expression in them are correlated, and histone modifications within the TADs are also correlated. Histone modifications are things which determine whether things are expressed, things are not expressed, and so they're regulatory elements. And the goal of this exercise is to identify and distinguish permanent and, I'm sorry, I skipped one critical point. Some are believed to be conserved across different cell types and even species. And our goal is to identify and distinguish between what we might call permanent TADs and transient TADs. Now, transient TADs are not inactive, but they're special to that particular cell line. So different groups of genes are active in different cells. And the TAD boundaries, as I mentioned, are physical insulators, which reduce interactions between elements on either side. And there's something called CTCF sites, which again is a regulator. And they act as anchors at the base of these loops. And so you look for TADs. If you look for TADs, you should be looking for things which have CTCF sites on either side. Remember, this whole thing, as you'll see, we have to imagine it as a line, right? I mean, it's been folded, but it's a line. And so each of these loops corresponds to an interval on the line. And the CTCF sites, CTCF sites mark the border, but not all CTCF sites are boundaries. Okay. Well, here's a description of the process. I'm not going to spend too long on that because I don't really understand it. But it's, it's a schematic of how they do these experiments. So they somehow do this business of cross-linking, and you can cross-link if things are close to each other. So that marks the points in the genome which are close to each other when it's folded. So they cross-link it, and then they cut it with restriction enzymes, which I think most of you have heard of. Then they mark the ends of these restriction enzymes. Then they form a loop. I mean, of course, when I say they do that, of course they don't do that for one. They do that for lots and lots at the same time. These are all chemical things. They ligate, and then they cut again. They purify and shear. They pull down the biotin, which means they basically arrange it so that you can actually identify what the letters are on the strands. So these are actually paired, they're paired strands. They're these two pieces. Oh, and of course I should have said that these are the strands of a single one of the molecules, right? So this is a chromosome. Each chromosome is a double helix. So the 2 strands per chromosome. Okay, and now, so as you can see, these experiments are quite recent, and they've gone up from doing it for single loci, trying to find out where they have contact on single loci, to sets of loci, to genome-wide, which is high C, and that's the data that we're going to be looking at. And of course, what we get from this sequencing is a proxy for measuring the 3D distances among all possible locus pairs in the genome. This work, by the way, I should mention, was first done, I believe, by Joop Dekker, who's from the Netherlands. What?
I have good genes.
Yes. So, okay. All right, so what do you get? Doing it base by base is, of course, much too precise. You bin it, and I would think I forget how many kilobases. And then you count in each fragment that you have, right, how many things have been pulled down. And that's an indirect measure of the contact. And so you make a matrix, let's say the raw matrix. You put the same chromosome, so this here simply lists the bases in order from 1 to whatever it is. And this is the same chromosome. And this anti-diagonal corresponds to the points which are, Do I have it right? I always get confused with this thing. Oh yeah, these things are so being far from the diagonal means that you are far from, positions are far from each other. And if you look at the diagonal, you'll see these things, sort of areas of concentration, and these things can be thought of as corresponding to TADs, and these maybe are boundaries of the TADs. They're far from each other. They don't connect. Note also that there are TADs within TADs. But the model that I'm going to talk about actually, at least initially, doesn't do that, doesn't assume that, doesn't permit that actually. So already, as I said, this is a very hot area. I think the data began coming in around 2012, and there have been lots of methods which were quickly thrown down to deal with these matrices. Oh, I forgot one thing, maybe just let me go back. It's not really critical. This data has been processed. This has been processed so that this matrix has uniform marginals, right? If you view this as an intensity matrix. And the reason for that is because you assume that each one should have the same exposure. Otherwise, you get biases of different sorts. Again, this is not critical for us. We deal with the matrix as it is. Okay, so there have been ad hoc methods for detection, right? The first one is just said, well, okay, this is actually a single string, and then there are these pieces, which are the TADs, which are different, and so you form an HMM and you try to distinguish when do things happen. But this doesn't take into account the fact that you have nested domains. You have to form a 1D summary statistic, so it's a fairly primitive method. Then there's a bunch of methods which involve somehow looking at the matrix and contrasting blocks in order to find out where the TADs are, right? Taking into account the two-dimensional structure. But again, all fairly ad hoc, not really model-based, and the number of TADs is not specified. And then finally, of course, now since we have network analysis, there has been some attempt to do block models on these things, where the bins are the nodes and the interactions are the edges, and then you do the usual thresholding. There is, however, this is pretty, you don't have the contiguity requirement satisfied. So you can have, you don't necessarily see a TAD as a single guy. You may see pieces, some of them phony. And the exchangeability assumption is unreasonable since the positions, it's again really the contiguity issue. That means that you cannot simply permute the indices and expect to have the same kind of probability distribution. So finally, I'll come to our model, which is a network model, but it has spatial constraints and biological covariates. And so I'll go to that next. And this is, as I said, there are no theoretical results. This is a model which is designed for this situation. But it has some points of contact with things that have been talked about at this workshop. So the basis, the abstraction is, of course, as usual, the matrix of frequencies. You then threshold them, and then we'll look at how stable the thresholds are to obtain an adjacency matrix. And you also observe, of course, a vector y, which tells you if you have a CTCF site or not. And I here is position on the univariate genome. Okay. So here's the model. The first assumption is, I think, reasonably well biologically founded, although again, as usual, there are exceptions. But all TAD boundaries are CTCF sites. So we only need to look at the CTCF sites. This one is not true, that there are no nested TADs. But in fact, what we propose, as you'll see, is a hierarchical procedure. You find the strong domains, and then you look within the strong domains to find subdomains. You know that they're not overlapping domains. At least that you know. And now you form a model. You have you take the thresholded frequency matrix, X. Oh, sorry. You form a new matrix, X. A is the incidence matrix. And this matrix is also M by M, where M is the total number of CTCF sites. And so these are actually the guys you want to find. You view this as the matrix of latent, unknown positions of TAD corners, TAD boundaries, right, which become corners. So here's you can see XAB is here. And so XAB is 1 if both YA and YB are CTCF sites, and AB is a TAD, which of course we don't know, and 0 otherwise. And then you tell yourself that the incidence matrix, given that you have a TAD, that you are in a TAD, right? So I and J are in the TAD, right? The TAD is AB, the interval AB, and I and J are in the TAD. Then the probability is high in some sense that probability that you have a good contact is high. And otherwise, right, you are in the background, and you make the probability beta. Notice that this is a model which has not that many, which has actually a fair number of parameters, right? If the number of TADs is large, but if the number of TADs is fixed, is not bad. On the other hand, you have latent variable for each possible pair of positions. So the goal is to infer the latent positions, of course the latent positions, the XABs, and the parameters, and then you iterate within XAB, as I said. I'm not going to talk about that. Questions on this?
Why would you do the bound? So why do you do the cutoff on the count matrix?
You mean why you threshold?
Why you threshold?
Because it makes the model more convenient. I mean, that's of course the standard technique in most network models. I actually agree with your implicit question: is aren't you throwing away information?
Yeah, but you do a logistic or something. I don't know.
Well, it's possible, but I don't know. Anyway, we maybe we were too steeped in the network world, but the results we got were reasonable, and they were reasonably stable to the threshold. And there's so much noise in this kind of data. So it's okay. So then we write down a likelihood. Oops. Let's see. Yes, likelihood first. So A and Y are what you observe. X is latent. Theta are the edge probabilities. You write down the log likelihood. It looks complicated, but it actually isn't too bad. You sum over all possible positions, which means, of course, all possible pairs of positions in the matrix. And so this says you get something if A and B are CTCF sites. XAB is 1, so you're in a TAD. And the IJ that you're looking at here is in the interval AB, right? It's in the TAD. And then AIJ is going to be the incidence, 0, 1. And this is what you get because you assume, so you're assuming that the AIJs are independent Bernoullis, if you only knew this stuff. And however, they're independent Bernoullis alpha AB if you're in the TAD, and they're independent beta if you're not in the TAD. So this part is simply the Bernoulli likelihood, but for being in the TAD, and this part is the contribution from the outside the TAD. I'm sorry. Oh, it has. All right. You mean I wasn't shouting loud enough?
It's okay.
It just flipped off a minute ago.
Okay.
But we heard you.
Yeah, exactly.
Maybe the people on the web right now may not.
Okay, fair. There are millions.
I see the batteries right there.
Live stream.
I'm not going to
Of
make
course, I may have
any-
inadvertently flipped the switch, right? Maybe it should be clipped just to make sure that I don't do that by moving. Okay, thank you. Yep. Okay, does it work? Right. Fine. Okay, so that's the likelihood, but then you have these side conditions, right? Because you don't know how many TADs, so you simply, this thing is parametrized by K, which is the total number of TADs. And then this condition is basically telling you that you have at most one TAD covering a given CTCF site, which you know isn't true, but you think that they're TADs of different orders of different powers, if you want. And now you want to you could maximize the likelihood of the constraints, but what actually turned out to be a bit better, and now we have some connection with the lecture in the first workshop that I gave. Actually, we use a variational likelihood. So, again, I always like the motivation for variational likelihood. I gave it in the first workshop. Some of you haven't seen it. It's actually somehow in the presentations of it somehow gets mixed up with exponential families, and it starts to get complicated. The idea is actually very simple. It's basically the following. You have a situation where you have, as we have in this case, hidden latent variables, right, and observed variables. And so what you're interested in is the logarithm of the observed, probability of the observed. Now, of course, that's simply the logarithm of the sum over all possible values of the latent variables of the joint. The point is that the joint is simple, right? The marginal is the bad part. Because in some sense you've got a sum over all possible values of the XABs. So that's actually the reason you don't want to do the straight likelihood. But, okay, so all you do is a simple trick. You simply divide and multiply each of the PAZs by an arbitrary probability measure on Z. And when I say Z, I don't mean individual Zs. It's the joint distribution of all the Zs. Okay, well, by Jensen's inequality, you have this. So you always have this is always a lower bound to the likelihood. And actually it's assumed. And it's assumed for psi star, which is simply the conditional probability of Z given A. Well, you say, wow, but the trouble is, that's the problem. The probability of Z given A is as hard to get as the marginal probability of A. However, what this suggests and what the physicists do routinely is that this is true if you look at all probability measures, right? So you have the statement which is correct. This is the max over all probability measures. But you can approximate. You don't know quite what you get, what the properties are, but in fact, in some cases, you can show that it behaves very nicely. In some cases, it doesn't. So maybe for those of you who are more familiar with it, if you take this thing to be a product measure, the approximation to be a product measure, this is the so-called mean field approximation. If you take the thing to be a product of functions of pairs of variables, which looks innocent, but of course creates horrible havoc because it then makes marginals, not so easy. Then you have what's called the Bethe approximation, or part of what's called the Bethe approximation. And then actually one can go further. In principle, one can go all the way to look at the things which are marginals on all of the n variables, which, of course, I mean, joint distribution on all the n variables, which of course brings you back to where you started. I mean, the trouble is that this probability is certainly not a product, or doesn't at all. But it's not actually so bad. Okay, and the set we take is, of course, simply a product, but where the parameters satisfy the side conditions, right? You basically take each of the XABs and replace it by a pi AB between 0 and 1, and you get this problem. And the nice thing about this problem is, well, there's several nice things, as you'll see in a second. But let's see, what did I? Well, so you maximize that. Oh, yes, I was going to say, I think for those of you who are here for the first workshop, which is probably a small subset, in fact, for example, for block models, it turns out that if the degree, average degree, goes to infinity faster than log n, the variational likelihood gives you the right answer asymptotically. And the reason is that that you can find algorithms which basically identify each of the latent variables, each of the XABs correctly with probability tending to 1. So that's a product measure. You have point mass on each coordinate. Whether that happens here too, we don't know yet, though I think that it's, and you know what parameters we have to look at, I don't know. All right, so there are various nice things about the algorithm here. One is that you basically go back and forth. The alpha parameter, which is part of the theta parameter, if you fix the, you actually can estimate it, if you want, in a simple way, without doing any iteration. And I actually don't quite. Oh yes, of course. That's simply it's a frequency that you're counting, and this is. Remember, you assume that you know. You're in a situation where you assume that you know that you're in a CTCF in a TAD, and if you're in a TAD, of course, the frequency is just counting the Bernoulli trials with probability alpha AB within the TAD. And then for a given pi, the maximum of L pi beta has a closed form, which I haven't checked. But presumably, again, you use the fact that if you're out of the TAD, you know what's happening also. And then it turns out that for a given beta, L pi beta, which is the thing which depends on pi, is convex in pi, which is not, unfortunately, the case in block models. Block models just give you lots and lots of false extrema. This is convex, I mean, partly convexly. Why? Because if you look at the variational likelihood, one part of it is an entropy, or minus an entropy, which is automatically convex, and the other part in this case is linear. So, okay, so you can really, it's reasonably fast. And if you can drop the entropy term, it's really fast, because then it's all linear. Now you might ask, how can you drop the entropy term? Well, remember that what you're looking for, actually, in the end, is point masses. For that, the entropy term is 0. So at the extremum, the entropy is 0. So it's not clear that that doesn't kill you if you start out, away from an internal point, but at least in practice so far, it works. Okay. Now, there's still left, as usual, the difficult choice of the upper bound on the number of TADs. But you can estimate it for any given k, which is a parameter of the optimization problem. And you can estimate it by simply the sum over AB of the pi hat ABs that you're getting, you know, the things that you've identified as TADs. And then if you plot that against K, lo and behold, you actually, I mean, here I'm reporting what my collaborators tell me, that you actually get a very nice elbow. And so that you can nail the number of TADs from this reasonably well. Now, as I mentioned, the big interest is not only in identifying the TADs, but also in separating between TADs which are transient and TADs which are permanent. Right? TADs which are present in all cell types somehow are special. And for example, you'll see later that we use a very weak test. Our collaborators promised to actually run some experiments which will give us a much stronger test. But intuitively, at least, but now it's me speaking, and it may be completely wrong, you would expect somehow things like housekeeping genes and so on, maybe, to be on the permanent TADs, you know, the TADs which appear in all cell lines. Functions which are general to cells should appear on one. But whether that's true or not, I have no idea. Well, it's very easy, of course, to modify this model, because if you have data from a whole bunch of cell types, since these observations are clearly independent, they're basically different experiments, you simply use the CTCF sites which are common across all those cell lines, right? Because that's what you're looking for, TADs which are common. And then you just form the likelihood, the threshold of frequency matrix from cell type T, and the number of the Y is taken from here. And then you have the same problems before. Now this, I have to admit, is ad hoc, and I can't say too much about it, but you make a further check on whether you haven't picked up things which are possibly phony, and the further check is that you do a test whether the interior of a TAD is really different from the exterior, and you do a Wilcoxon test. This, as I said, is ad hoc. It does, unfortunately, have an effect. If I could tell you that it does nothing, then I would.
It has a positive effect from a scientist's point of view.
Yes, it has a positive effect on the side. It cuts down on the number of TADs, but you might, it shows that the model is an imperfect approximation. Okay, so we actually have, this data is from a published paper, Rao et al. In Cell, and that's why you'll see the comparisons we make are with the TADs that they've identified by one of these ad hoc methods. However, we have access to, and that's the reason we started this whole enterprise, to a whole bunch of new data, which is being produced by Kundaye and other labs at Stanford, which we will apply this to. Okay, and so what you see here is simply there are 5 cell types. It's high C data, so it's for the whole genome, which means the scale is big. And that was actually, of course, one of the issues why this took so long, is implementing it. It's only now ready to be applied to these larger data sets. And you look at 3 levels of thresholding, just to try to stabilize. And then you compare with these arrowhead domains, which were obtained by an ad hoc method in this other paper. So the trouble is, as is often the case in biology, that we don't have ground truth. Right? Nobody has gone in and checked these TADs individually. But there is a general belief, though not all agree, that you would expect to have a concentration of histone marks near the boundaries of the TAD. Right? And I don't know what the real basis for that is, but everybody does that. So that's a test. And we made a comparison between the number of domains that we found. These are cell types, right? Number of domains found by us and the number of domains found by Arrowhead. And for this slide and this slide, it's fairly clear that we have a higher concentration in our domains, right? And also, the number of domains that we find is somewhat more stable.
Do they find larger domains? So basically, so when you look at the triangles, do they pick up the bigger triangles, or they just pick up fewer triangles?
They pick up fewer triangles. Well, actually, I think, oh, damn, I'm not sure I brought that slide. They pick up some of the same triangles, they pick up some different triangles, and yeah, I guess in some sense they do. The triangles, since they have fewer, I think they do get bigger ones, but I'm not sure. I don't. Okay, so this is, unfortunately, this is another cell type, and this time they're doing better, but they have more domains. They consistently have, but then here they have fewer domains, and we do better on the whole, and here we do better consistently. So on average, our method is better on this rather weak measure, right? So, but we have taken into account things that they haven't. For example, the contiguity. We don't get any broken domains. And then stability. Well, thresholding at the 75th quantile versus the 80th quantile. We found 73 domains at the 75th, and I guess the same number with the higher index, but 53 of them had a very high Jacquard index, so they're really evidence of being the same domain, right? I mean, and the other evidence is much the same. Here you check replicates. So this again stability. There are two replicates of the same cell line. And as you can see, again, in one you find 70, in the other one you find 69, and 60 have a very high Jacquard index. So actually I find this the comparison which is most satisfying, that you really do have stability, or some evidence. I'm not going to go into the data normalization thing. Here's a picture. So these are the domains that you find using all 5 cell types. And these are the domains that you find using individual cell types. And in fact, and then this was obtained by there was some iteration, I think. In fact, this almost seems to be, to me, crudely, a union of the others, which is I think they sorry, I'm not sure actually whether all 5 cell types means they pool the domains, or whether all 5 cell types means that you use the likelihood for all. I mean, it must be that you use the likelihood for all. Okay, then, all right, so now you do joint inference. You have 73 domains, 22 of which have a high index with at least one domain in every cell type. After all, something which you claim is present in all cell types should also be present in the individual cell types. Again, the trouble is that you can't fix the boundaries that well. You always have to check with the Jacquard index. But here's some other biological evidence. So these are histone modifications, different histone modifications, and the tracks that you get in the different cell types. So these are cell types. And they seem to match pretty closely. And you expect the histone modifications, remember, to be near boundaries. So this suggests that you're finding, again, another evidence that you're finding similar you're finding the same boundaries. This is for one, of course, of the 22 domains, which have a common in some, like with the hydrocard index, with something in every cell line. Okay, so that's what this is. It's actually a single big domain. And then you can get average correlation, which is between histones in different cell lines in the 22 domains versus random domains. And you obviously get the random domains are all over here, pretty much, and the others are shifted. So I think I'll just skip this too. Ongoing work, and this is why I would have liked to present some theoretical guarantees. I believe it's not very hard to show that if you fix m and k, sorry, if you fix k and you let m go to infinity, then that is the number of CTCF sites, you get consistency with our algorithm. It would be nice to have an automated way of choosing the threshold. It would be nice, we would want to work on a model of the changes across cell types. I find the last two, of course, being the most important, getting further results from more chromosomes, and finally, functional validations of the conserved TADs. And unfortunately, we can't do that. Our collaborators have to do that. So, what does this show? It shows that you can write a network type model in situations like this, that you get results which are reasonable, and we hope will be validated. It's also an illustration of how the mean field technology goes. It's not limited to block models, but can be used in these more general situations. And that's it.
Okay, so questions? Yep, Karen?
Yes.
Go ahead.
Yeah.
Okay.
Very interesting
Jizzy.
work. I've been trying to think about the stochastic block model and how your model fits in. So in the stochastic block model, when you've got a sparse network, people often now use the degree-corrected stochastic block model. So
Yes.
somehow correct for the degree. And I wonder whether your YAYB do that, or whether you've got any way of...
No, I mean, it's different from the stochastic block model because really we're using, in this likelihood, if you think about it, we're using very much the position of the bases, which, of course, the stochastic block model can't do because it's an exchangeable model. And that's the big difference. I mean, yes.
But you still have an underlying adjacency
I'm sorry.
matrix. You still have an underlying adjacency matrix, of course.
It is an adjacency matrix, all right, but with an adjacency matrix, we know if we are using a stochastic block model or any exchangeable model, we know that the labels that we're putting on the nodes are arbitrary. I could permute that matrix, and it would be just as good, just as good an adjacency matrix. Use spectral methods on that, you get the same thing, right? Whereas here, that's not true. That's not true. The positions do mean something, and that's why I think just using standard, any model really, coming from standard methodology would probably doesn't give
But
sense.
I agree on that. I just wondered whether there's something like a degree correction, which might get better results.
It's possible. That's possible. We haven't thought about that. I think actually, I have to admit that I would like to think, following Ernst's first question, whether one couldn't try to use the raw intensities themselves, although, as you can see from the experiment, things are really very noisy. Right? I mean, this is a hard experiment. Well, you can't see, I just gave you a schematic, but I can assure you that it's a hard experiment. The reason it's only been done so recently is also
Just to follow up on that, so basically, if you look at the stochastic block model, basically the additional thing here is the diagonal, right? That's kind of destroying the us first, maybe call it the first off diagonal. The fact that neighbors are very highly
Right, and I mean,
have high counts next to each other.
that's right.
That's why it's not a stochastic.
That's right. That's right. Well, I mean, and it's not just neighbors. I mean, it's there's
Yeah, and then it's
it keeps on going. I mean, there's
close things
there's
that's close
dependence.
by.
There's dependence. That's right.
Yeah.
Exactly.
Maybe then. Okay, Kerry.
Peter, on the consistency suggestion you had there, that you get that. I mean, it seems to me that the nested structure, though, so I know for your model there's no nested. But
We haven't been using the nesting yet. Yeah.
right, but in the nestedness of the real data might really break that kind of result, right?
Yes, that's right. That's possible. I mean, I think it's a question again of the strength of the. It probably depends on the strength of the parameters. But you're right. I mean, if you had sort of a TAD which had a lot of little TADs, yes, I think might very well break it.
Yeah, I mean, you might not really be robust to certain.
Yes, that's true. That's true. Again, part of it is that in some ways I think my collaborators have not been working fast enough because there's so many people working in this area. On the other hand, they've been working as fast as they can. and we certainly would want to build in, that wasn't mentioned but should be, that I think you can build in the hierarchy in somewhat the same kind of ways that you do. Yeah.
Questions? Yep.
Simon.
Peter, just to make your life more miserable, this is now done at single cell resolution.
Well, does that...
Noise is even worse.
Worse, yes, that's right. Yeah, so in that sense, you make life more miserable indeed. Right, that's right. On the other hand, I presume, I mean, I have great faith in probably misplaced from given some encounters in the biologists, that the techniques are also getting better.
Oh, absolutely.
Yeah. So
Inside your models, you also assume that you know all the potential cutoffs, right? The potential CTS.
you assume that CTCF sites are, yes, that's correct. And that seems to be reasonably, the biologists all nod that that's a reasonable assumption. Of course, you may miss some because, as usual, there are some exceptions. But in their
This because in your likelihood, you could basically add a nice additional term, basically just letting your things, your boundaries vary. Or
yes, you could. That's right. But now you increase the number of parameters tremendously, right? The latent variables. And at least, as I may have told people when I gave the talk in the first session also, we found at least that variational likelihood, mean field likelihood, can break down quite badly as the number of parameters increases, because you have the whole function is not convex, and there are many false minima.
But you could post hoc do it like basically you, in the end, just adjust your boundary maybe by 1 or 2 or 3. Or
Sure. No, I think that's right. I mean, yeah.
Okay, further questions? Okay, if no further questions, then let's thank Peter again. Next talk is at...